Today, we are diving into a pretty amazing scientific quest. It's really a search for a better way to predict what the future holds for patients with cervical cancer. This is the story of how a team of researchers took a literal mountain of data to find a tiny but incredibly powerful clue hidden deep inside our own genes.
And make no mistake, this work is so important. Because cervical cancer is the fourth most common cancer that affects women across the globe. So, getting better at predicting its path isn't just some academic exercise. It could genuinely change countless lives.
You see, getting a diagnosis is really just the first step. The real challenge for both doctors and patients is figuring out what comes next. Is this particular cancer going to be aggressive? How well will it respond to treatment? Answering those questions means we need better and hopefully less invasive tools than what we've got right now.
So where do you even start looking for those answers? Well, the researchers behind this study turned to some of the smallest yet most powerful molecules in our bodies, microRNAs. These things hold incredible promise, almost like biological fortune tellers.
Okay, so what are they? The easiest way to think about them is like this. If your DNA is the master blueprint for your entire body, then microRNAs, or miRNAs, are kind of like the foreman on the job site. They run around acting like dimmer switches, turning the activity of certain genes up or down just to make sure everything is running smoothly. But here's the thing. miRNAs have two very different faces. In a healthy cell, they're the good guys, these essential regulators. But when their expression gets all scrambled up, they can totally flip a switch and start driving cancer. Sometimes they act like an oncogene, basically flooring the gas pedal on cell growth. Other times, they silence the tumor suppressors, the very things that are supposed to act as the brakes. And it's this dual nature that makes them such powerful clues for cancer detectives.
All right, but we have thousands of different mRNAs in our bodies. So how in the world do you find the exact ones that can predict a patient's future? Well, that's where some really serious data detective work comes into play.
So the researchers started with a true data mountain, a ton of genetic information from over 300 patients. First, they had to sift through all of it just to find the mRNAs that were acting weirdly in cancer tissue. That narrowed the pool of suspects down to 101. Okay, better. Then they asked the really critical question, which of these are actually connected to how long a patient survives? That brought the list down to 52. But even that's not enough. You need the right combination. So for the final step, they used some really powerful modeling to find the absolute smallest, most predictive team of mRNAs, the core signature.
And after all that filtering, after climbing that entire mountain of data, it all came down to just these three. That's it. A compact, incredibly powerful signature made up of 3 specific microRNAs. When they act together, these 3 little molecules hold the key to predicting a patient's risk.
But, finding the signature is only half the battle. Any good detective has to check their work, right? So the next step was to put this 3-mRNA model through some really tough tests to see if it could actually do what it was supposed to do, predict patient outcomes. And wow, the results were pretty striking. On this survival curve, that top blue line represents patients the model flagged as low risk, and the bottom orange line is the high-risk group. Just look at how quickly those two lines pull apart. That big, clear separation shows that patients in the high-risk group had a significantly worse prognosis. The model was successfully sorting them. We can also put a letter grade on that accuracy. This chart shows what's called an AUROC score, and it just measures how well the model can tell the difference between high-risk and low-risk patients. A perfect score is 1.0. For predicting 1-year survival, this model scored a 0.84. That's a really strong result. It shows it's highly effective, especially at flagging patients who are at more immediate risk.
Now this is like the final exam. The researchers took their model and tested it on a totally different set of patient data from a different study. And look, it worked again. It still successfully picked out the high-risk patients. This is so critical because it proves the model is robust, that it wasn't just a fluke that only worked on the original data. Okay, so now we know the mRNA signature actually works. The next logical step was to see if they could combine it with other clinical information, to create an even more powerful, practical tool for doctors to use.
So the researchers looked at all the usual clinical factors, and they found that besides their new miRNA risk score, one other very familiar factor was also a really powerful predictor all on its own, the clinical stage of the cancer. You can see here that both that risk score and being at stage 3 or 4 are really significant.
So by combining those two power players, the new mRNA score and the cancer stage, they created this, a nomogram. I know it might look a little complicated, but it's a beautifully simple and powerful tool. A doctor can just find a patient's risk score on one line, their cancer stage on another, draw a line down to the total points, and from there, get a personalized probability of 1 year and 5 years survival.
And maybe the absolute best part of this whole story is that this isn't just some theoretical idea locked away in a research paper. The scientists actually built a free, publicly available web app called CESCProg. They put this powerful tool right into the hands of researchers and clinicians anywhere in the world.
I mean, it's just a fantastic achievement that really moves us closer to more personalized medicine for cervical cancer. And it leaves us with one final, really exciting question to mull over. If this kind of data-driven detective work can decode the future of one type of cancer, what other diseases could be next?